Neurulation.
We are just going to discuss about the formation of neural tube, and there are two ways in which the neural plate is converted into a neural tube. In the primary neurulation, the cell surrounding the neural plate directs the neural plate cells to proliferate, invaginate and pinch off from the surface to form a hollow tube. At the same time, in the secondary neurulations, what happens is the mesenchyme cells, they form a solid cord and subsequently forms a cavity. Process is different in different vertebrates, and if you just say the bird, the neural tube anterior to the 28th stomoid pairs is made up of primary neurulation. At the same time, in mammals, the tail region, in amphibians, the tail neural tube is derived from the secondary neurulation. The same pattern is also observed in the fish. Events which occur during the primary neurulation process involves internal positioning of the neural tube, which will form the brain and spinal cord, external positioned epidermis of the skin, and neural crest cells. These neural crest cells, they form in the region that connects the neural tube and the epidermis, and they migrate to the new locations where they will generate the peripheral neurons and glia, the pigment cells of the skin, and several other types. The process of primary neurulations appears to be similar in all the vertebrates, and after the neural tube formation, the edges thicken and move upward to form a neural float, which is a U-shaped neural groove which appears in the center of the plate, dividing the future right and left side of the embryo. Shows the derivatives of ectoderm. Since the derivatives of ectoderm include the surface ectoderm, neural crust, and the neural tube, and various organelles and organs, they just differentiate or derived from all these three crust cells. During primary neurulations, the neural folds migrate towards the midline of the embryo and eventually they fuse to form a neural tube beneath the ectoderm layer. So the cells at the dorsalmost portion of the neural tube becomes a neural crust cells, and there are distinct four phases in the primary neural formation, and each one they just overlap with each other. And first one is formation and folding of the neural plate, and second one is shaping and elevation of the neural plate, and then convergence and creating a neural grooves, and closure of the neural grooves to form a neural tube. Formation and shaping of the neural plate. The process of neurulation begins when the underlying dorsal mesoderm signals the ectoderm cells above it to elongate into a columnar neural plate cells. So these elongated cells, they form the flattened pre-epidermal cells. Usually half of the ectoderm is included in the neural plate. These neural plate, which has been shaped by the movement of the epidermal and neural plate regions, they lengthens along the anterior posterior axis and narrows by the convergent extension, and they intercalate several layers of the cells into a few layers. These convergence and extension movements are critical for the shaping of the neural plate, and the mutation disturbing this movement can block the neural tube closure. the neural plate or preferentially in the anterior posterior direction. Example in chick, often it is in rostral caudal or beak tail directions. These events occur even if the involved tissues are separated. If the neural plate is isolated, its cells converge and extend to make a thinner plate but fails to roll up into a neural tube. However, the border regions containing both presumptive epidermis and neural plate tissues is isolated. It will form a shows how the two sides of the hinge regions they get folded to form a groove. Phase in neurotive formation is a bending of the neural plates, which involves the formation of hinge regions where the neural plate contacts surrounding tissues. Especially in birds and mammals, these cells at the middle of the neural plate forms the median hinge point. And the cells for this median hinge point are derived from the portion of the neural plate just anterior to the Hen's node and from the anterior middle line of the Hen's node. These cells becomes anchored to the notochord beneath and then form a hinge which forms a furrow at the dorsal midline. The notochord induces these median hinge point cells to decrease in height and to become a wedge shaped. Cells lateral to these do not undergo any such changes. Shortly thereafter, two other hinge regions forms furrow near and connect at the neural plate with the remainder of the ectoderm. Two regions, that is dorsolateral hinge points, and they are anchored to the surface of the ectoderm, and they increase the height and become the wedge shape. And this wedging is intimated linked with an microfilaments and microtubules which are both involved in these changes. Actually, when an inhibitor called chisin of the microtubule formation inhibits the elongation of this shell formation, while cytohalastine B, an inhibitor of microfilament formation, prevents the apical formations, that is an wedging formations. As the neuroepithelial cells enlarges, the microfilaments and their associated regulatory proteins get accumulated at that point. This constructions of this microfilament changes the shape of the particular area. After the neural groove formation, the closure of the neural tubes occurs. The neural tube closes the paired neural folds as they are brought together at the dorsal midline. These folds adhere to each other and the cells from the two folds merges. In such a case in birds, neural crystals do not migrate from the dorsal region of the neural tube until and after the neural tube has been closed at that site. In mammals, these cranial neural crystals migrate while the neural folds are still being elevated, whereas spinal cord regions, the neural crystals do not migrate until the closure has occurred. The closure of the neural tube does not occur simultaneously throughout the ectoderm. This phenomena is best seen in the vertebrates such as birds and mammals whose body axis is elongated prior to the neural. Last chick embryo, neurulations in the cephalic, that is head region, is well advanced, while the caudal regions of the embryo is still undergoing gastrulation. The two open ends of the neural tubes are called as anterior neuropore and the posterior neuropore. Chick neural tube closure is initiated at the level of future midbrain and the zip up in both directions. In human, there are probably three signs or more signs of neural tube closure. The failure of this closure in different regions of the neural tube causes different neural defects such as they induces, that is the day around 27th of development results in the condition called spina bifida, and the regions which has been depends upon the spinal cord remains exposed. Failure to close at the site of 2 or 3 in this rostral neural tube Entire neural tube to close over the entire body axis is called as cranioacryscysis. Collectively, neural tube defects are quite common in the humans, that is, it occurs in 1 in every 1,000 live birth. The neural tube eventually forms a closed cylinder and separates from the surface ectoderm, and this separation is mediated by both environment and then genetic factor. As the cells which are going to develop into neural tube, they express the E-cadherin, and they stop producing this protein as the neural tube forms, and instead of that they secrete N-cadherin and N-cam. The derm and neural tube tissues no longer adhere to each other. The surface ectoderm, which was been experimentally made to express N-cadherin by injecting the mRNA into one cell of two-cell stage xenophus embryo, the separation of the neural tube from the presumptive epidermis is dramatically impaired. And in human, this has been the closer has been interplayed between the genetic and environmental factors such as the expression of PAS3 sonic hedgehog. These are the important mammalian neural tube formation genes, and the dietary factors such as cholesterol and folate. And it has been estimated that more than half of all the human neural tube birth defects could be prevented by the pregnant women which is taking a supplement, and it has been recommended to take zero point 0.4 milligram of folate daily. Relations, and in the next class we'll be discussing about how the brain get developed from the neural tube. Thank you. This shows the surface ectoderm where, and below that you can see the condensing mesenchymal cells, and after that they form a medullary cord, and there is a transition region between the anterior and posterior, and it forms a groove and neural tube formations, and below is notochord. Material for both neural tube and mesoderm. In human and chick embryos, there appears to be a transition regions between this junction of anterior and posterior, that is between the primary and the secondary neural tube. In human embryos, and these cavities are seen in the transition regions, but the neural tube also forms by the bending of the neural plate cells. Some posterior neural tube anomalies results when the two regions of the neural tube fails to relations, and in the next class we'll be discussing about how the brain get developed from the neural tube. Thank you.